Okay, let's move on to the next question. Okay, question 5. Some athletes train in high altitudes to improve their performance. Performance improves if more energy is released from aerobic respiration. So part 1 says complete the balance symbol equation for aerobic respiration. Okay, let me just bring out the pen. Okay, so aerobic respiration is glucose plus oxygen, and that would give you carbon dioxide and water. The formula for glucose is C6H12O6, and the formula for water, that's H2O, and if we balance it out, it'll be a 6 here. The reason why is there's a 12 here for the hydrogen, so there must be a 6 before the H2O. So that's balanced now. And then for part 2, aerobic respiration produces ATP. Describe how energy is released from ATP during exercise. So ATP basically gets broken down. So we would say ATP gets broken down, or you can say gets hydrolyzed. That's fine too. To form ADP and an organic phosphate, and you can write that as Pi. All right, on to the next question. So that is part B. So the table shows the percentage change in mass of hemoglobin in the blood after different periods of time training at altitude. So basically what happens when we train at high altitudes, there isn't a lot of oxygen at high altitudes, and so our body tries to compensate by producing a lot more red blood cells, which ultimately leads to an increase of hemoglobin, which is what we see here, right? So the longer you train, the higher the mass of hemoglobin or percentage change in mass of hemoglobin is. So let's plot that on the, what's on the other page? Okay, this is going to be a little tricky. Okay, so the x-axis would be the period of time in hours because that's the independent variable, and the y-axis would be the percentage change in mass of hemoglobin. So the largest number for this is 600, so I'm going to have to split this into 600. So 1, 2, 3, 4, 5, 2, 3, 4, 5, 6. Okay, so this should be 0. This is 100, 200, 300, 400, and 500, and 600. That's just enough. And don't forget to label the axes as well. Let me just double check. So period of time in hours. Period of time, and I'm just going to write brackets, hours. Okay, now let's check the y-axis. So your y-axis is your dependent variable. It's the percentage change in mass of hemoglobin. So the highest number here is 7.2. Let's see if we can try and fit that. So maybe 1, 2, 3, 4, 5, 6, 7, 8. Okay, so we'll have it so that it goes like this. 1, this is 2, this is 3, 4, 5, 6, 7, and 8. Don't forget to label the axes. So that is percentage change in mass of hemoglobin. So percentage change of mass of hemoglobin. Okay. So we're supposed to plot the results on the grid and then draw a curve of best fit. Okay, so the first point is 0, 0. So that'll be all the way here. I forgot to write this down. 0, okay. So 0, 0. The next one is 100 and 0.7. So 100 and 0.7, so it's 987. Yep, so we draw a little X there. Next we have 202.4. 202.4, that should look like this. One thing to note is that make sure that when you draw out the graphs, you don't do a little dot like this. Whoops. You don't do a little dot like this. They would usually prefer you to draw an X instead. Okay, let's move on to the third point. That's 304.1. So 304.1, that's there. This one is 405.9. 405.9 is here. Okay, and then we've got 506.5. 506.5. Okay, and lastly, 607.2. 607.2. Another thing to note is that you want to make sure that your plot takes up more than half of the space that's given to you. So this should be fine. And then the last part, we need to draw a curve of best fit. So let me just zoom out a little bit here. All right. And the third part, discuss how the percentage change in mass of hemoglobin after training at altitude could result in an increase of performance during exercise. So let's look at the trend here. So we can clearly see that as the time of period increases, the mass of hemoglobin also increases as well. So we need to write out the trend here. So we can say as the period of training increases, the of hemoglobin increases. So this is because more, why does that disappear? This is because more red blood cells are being produced. So hemoglobin is responsible for delivering oxygen to the body and therefore more oxygen can be carried to the muscle for respiration, or I should say specifically for aerobic respiration, to release energy. Okay, let's go through the marks. So 1 mark for as the period of training increases, mass of hemoglobin increases. Another mark for there are increase in red blood cells. Another mark for hemoglobin, it basically delivers oxygen or binds to oxygen, and therefore there are more that are being transported to the muscle. That's another mark. And then for aerobic respiration, that's another mark. To release energy, that's another mark. Okay, let's move on to this part, question 6. So I'll give you guys some time to read through it and we'll go through the questions. Okay, part 1. Describe the benefits of producing iPS cells from a patient's own cells. So because you're using the patient's own cells, they're going to be genetically identical to the patient. And because they're genetically identical, the antigens, which are the little proteins that are found on the cell membrane, they are not going to be recognized as foreign by the immune system. That's a great thing because then these cells do not get rejected by the patient. So another advantage of using iPS cells is, you can see this in the second paragraph. So it says, in the past stem cell research, scientists needed to obtain human embryonic stem cells from embryos produced by IVF. So if you use iPS cells, then you wouldn't really need to use human embryonic stem cells for your research then. So let's just write that down. Let's talk about the rejection first. So the iPS cells will have the same antigen as the patient, therefore rejection will not occur. And then we'll mention the other point, which is no longer needing to use the human embryonic stem cells. So another benefit do not need to use human embryonic stem cells. Okay, so one mark is the not needing the human embryonic stem cells. The other mark is same antigen. Another mark is rejection will not occur. Probably will not occur is quite a strong phrase. I might just change that so will less likely to occur. Less to occur. Okay, let's go on to part 2. So give 2 possible risks for using stem cells to treat diseases and medical conditions. All right, so let's go back up here and see if we have any information that we can use here. So if you look at the last paragraph, it says, this technique is easy to replicate, but there are concerns that unlicensed clinics are offering stem cell treatments. So that's probably one possible risk. So another thing about stem cells is there's a chance of them developing into a tumor. And the reason why is because stem cells can over time accumulate a lot more mutations, and that could lead to tumors. So you can say it may All right, let's look at part B. One condition that stem cells could be used to treat is age-related macular degeneration, or AMD. This is the main cause of vision loss in people over the age of 50. The graph shows the number of people affected by AMD in some countries. So part 1, it says calculate the difference between the number of women and the number of men affected by AMD in the United Kingdom. So for this question, we're only looking at the United Kingdom. And let's start off with the women's first. So each of these lines represent 200,000, right? So then that means that this line here represents 800,000. And each of these lines, so if each square or each large square represents about 200,000, and there are 5 smaller squares in the large square, then each of these smaller lines, each of these smaller squares represent 40,000. So this would be 840,000, and then because it's halfway, that would be 860,000. So for women, that would be 860,000. Now let's look at men. So each of these lines again represent 200,000. So then this would be 400,000. Okay, now there are 2 lines there, so that's 480,000 for men. All right. Now it says calculate the difference, so you would just do this number, the larger number, minus the smaller number. And that will give you 380,000. Okay, let's look at part 2. The diagram shows a human eye. AMD affects the retina of the eye. All right, so because it affects the retina, Z is the retina, so it'll be this one. But let's go over these structures. So X would actually be the cornea. Y would be, so Y basically controls the length of the pupil, so that would be the iris. And W would be the lens, which helps refract light into the eyes. All right, part C. A deficiency of vitamin A can also cause blindness. A genetically engineered rice product can be used to prevent deficiency of vitamin A. Describe how genetic engineering can be used to produce this rice product. Okay, so I've included a diagram here that can help sort of visualize everything. So a lot of this is very term heavy, but I'll let you know the things that you do need to know. So number one, you need, bacteria have plasmids in them. And this is the plasmid here. So these plasmids are able to contain genes, and then we can insert these genes into the bacteria. So what we would do is we would cut both the plasmid and the gene for vitamin A with the same restriction enzyme. So restriction enzymes are enzymes that are able to locate a specific sequence and cut at that point. Then we would insert the plasmid into the bacteria, and then we would infect the bacteria. We would infect the plants of the rice with the bacteria to be able to produce that golden rice. So let me make this one smaller so that we can write this out. Okay, let me just zoom this in here. All right. So describe how genetic engineering can produce this product. So first, the gene for vitamin A and the plasmid are cut with the same restriction enzyme, then insert the gene into the plasmid and insert that plasmid into the bacteria. The bacteria can infect the rice plant. So that golden rice can be produced. Okay, so let's go through the points. So you would need to cut out the gene for vitamin A, so that's 1 mark, with a restriction enzyme, that's a 2nd mark, and then insert the gene into the plasmid, that's a 3rd mark, and then infect the rice plant, or you can say infect the rice genome, that's a 4th mark, and that can produce golden rice, that's a 5th mark. So let me just pull out the diagram so that you can see it again. So once the bacteria infects the plant, these plant cells are able to grow in a culture so that the entire plant would have the gene for vitamin A. Okay, let's move on to question 7. The renal system is involved in osmoregulation. Name the area of the brain that detects the water content of the blood. That would be the hypothalamus. Oops, okay. Okay. The kidneys are part of the renal system. Put 1 tick in each column of the table to identify the function of the different structures in the kidney. So at the bottom, I've just included a diagram of a nephron just to do a little brief recap. So here, blood will enter through what we call, maybe let's just write it down here. The blood will enter by the renal artery, and then it will exit via the renal vein. Okay, and then here the glomerulus is just a lot of capillaries, and what happens in the glomerulus is that fluid gets pushed out, so substances get pushed out. So anything that's small enough, things like water, things like salts, things like urea, things like glucose, things like amino acids, they would all pass through the glomerulus and into the Bowman's capsule. And then your proximal convoluted is where, that's this part here, sorry, let me just circle that properly. That's this part here. So the proximal convoluted tubule is responsible for selective absorption. So for example, because glucose is small enough to pass through, what the proximal convoluted tubule does is absorb as much glucose and as much amino acids as possible. So let's go through the different structures here. So first one, it says regulation of water level. So that part is more to do with your loop of Henle and your collecting duct. So none of this would apply, and your loop of Henle is this part here, and your collecting duct is this part here. The next part is ultrafiltration, so that applies to the glomerulus. So ultrafiltration, like I mentioned, is when the smaller substances get pushed out from the glomerulus to the Bowman's capsule due to high blood pressure. Next is reabsorption of glucose that happens in the proximal convoluted tubule, and that is selective reabsorption essentially. The third part is removal of blood from kidney, so that would be the renal vein. Supply of blood to kidney, that would be the renal artery. So removal of urine from collecting duct, this would be the ureter. If you don't know what that is, so if we have like the little kidneys, this is the bladder, let's just draw a triangle. This tube that connects the kidney to the bladder is called the ureter, and then the tube that connects the bladder to the outside world, that is the urethra. Okay, let's go to part B. The table shows the percentage composition of 2 urine samples taken from one person. Not all the components were shown. The first sample was taken at midday, and the second sample was taken 4 hours later. Okay, so the first part, it says describe how urea is produced in the body. So urea is produced by the liver, and that's when we break down excess amino acids into urea, and that process is called deamination. So urea is produced in a process called, sorry, let me write that as one word, deamination, which occurs in the liver. Okay, let's move on to part 2. The ADH response changes the percentage composition of urine. Explain the ADH response that occurred in a person's body between the first and the second sample being collected. So let's just do a little bit of overview as to what ADH does. So here we have an example of if we have too little water in our blood. So if we don't have enough water in our blood, our hypothalamus is able to detect that. And so that's this part here. And then our pituitary gland is responsible for releasing a hormone called ADH. So ADH actually stands for antidiuretic hormone. And so ADH gets secreted and into the blood and will reach the kidneys, and specifically it targets the collecting ducts. So what happens with the collecting ducts is that the collecting ducts become more permeable to water, and therefore more water gets reabsorbed. And because more water gets reabsorbed back into our body, less water is being lost as urine. And so therefore our blood water level returns back to normal. So let's just make this, actually let's go back to the table. So if we look at the table, we can see that there is actually less water in the second sample compared to the first sample. So because there's less water in the urine, then that means that more water is being reabsorbed by our body. In that case, ADH is actually being secreted here. So let me write that down. So we can say the second sample has less water, which means that the urine is more concentrated. This happens when a person has, why do I keep spelling it like that? Has less water in their blood. Therefore, ADH gets secreted, which causes the collecting duct to become more permeable to water. So that more water can be reabsorbed. Okay, Whew, that's a long answer. Let's go through the marks. So 1 mark for the second sample has less water, another mark for urine is more concentrated, a third mark for person has less water in their blood, a fourth mark is ADH gets secreted or gets produced. So that's 1, 2, 3, 4. Fifth mark would be becomes more permeable to water, or collecting duct becomes more permeable to water. And then sixth mark is more water can be reabsorbed. Okay, let's move on to question 8. When women cannot conceive naturally, they can have fertility treatment. Women having fertility treatment can be given the hormone FSH and LH. Give a role of LH in women. So the hormone LH stands for luteinizing hormone, and that is the hormone that is responsible for triggering ovulation. So it causes ovulation to occur. Okay, part 2. Giving birth to twins or triplets is described as multiple birth. Explain how the use of FSH to treat infertility could increase the chances of multiple birth. So I've included a diagram here that shows the maturation of the follicles. So you don't need to know too much about the nitty-gritty details, but FSH is the hormone that is responsible for the maturation of these follicles. So you can kind of see before they start off as primary follicles and secondary, and then the egg gets released. So if you have more FSH, then more follicles get matured, and therefore more eggs are produced. And if you have more eggs that are being produced, then that increases the chances of fertilization from with a sperm. Okay, let's make this one smaller. Okay, let's write this down. So FSH causes the maturation of follicles in the ovary. Okay. This leads to more eggs being produced, which would increase the chances of it being fertilized by a sperm. All right, 1 mark for maturation of the follicles in the ovary, another mark for more eggs being produced, and another mark for increased chances. Another mark would be increased chances for the egg being fertilized by the sperm. All right, let's move on to part B. So some identical twins share a placenta, as shown in the diagram. Twins that share a placenta are often born with a lower mass than twins that have separate placentas. Explain why twins sharing a placenta have a lower mass when they are born. So the function of the placenta is for exchange of materials. So nutrients would go from the mother to the fetuses, and metabolic waste like carbon dioxide would go from the fetus to the mother. Now, if they have one placenta, if they're sharing a placenta, then that means that they're sharing the nutrient supply that's coming from the mother to the fetuses. And if they're sharing the nutrient supply, then that means that they are getting less oxygen and less glucose, which means that they're undergoing less aerobic respiration, which means less energy is being produced by these fetuses. Okay, let's write that down. So, why would they have a lower mass? If our if they are sharing a placenta, they would and glucose from the mother. This would mean less is which means less growth occurs. Okay. Part 2. Identical twins form when the embryo splits early in pregnancy. Explain why the twins are genetically identical. They are genetically identical because they come from the same egg and sperm. And when that happens, a zygote forms, and then it forms into an embryo, and then this embryo would split into two. So they are genetically identical because, or this is because they originate from the same egg and same sperm, then the embryo splits into two. Okay, let's move on to the third part here. Part C, when babies are young, their growth is monitored using a percentile chart. The diagram shows a percentile chart for baby girls. Okay. Determine the growth percentile of a 6-week-old baby girl with a mass of 5 kilograms. Okay, so 5 kilograms, that's about here. Okay, and then 6 is about here. Okay, so that lines up to that solid line, and that would give me 75th percentile. Okay, last part, part 2. Describe how percentile charts are used to identify concerns about the growth of the baby. Okay, so it's very important to weigh the baby regularly and compare it with other babies their age as well to make sure that nothing wrong is happening with them, because if their mass is too high or too low, then that might be an indication of something concerning happening with the baby. So let's write that down. So the baby is weighed regularly to ensure that their mass is comparable to babies of the same age. If the mass is too high or too low, this could be a sign to that something be concerning. All right. Okay, so that is paper 2 done. Good luck for your exams.